Right, I have Tsepisolekwe, which I believe is Sutu, if I'm correct. Am I, guys? Okay, Khomotso, I can see him nodding there. Right, so Tsepisolekwe Khomotso. Thanks for your question. We are going to do your question, which means you're also in line for a prize this week. Rightio, so it says, a scientist gathered information about the affected and unaffected individuals for a certain genetic disorder. Sounds interesting. It's like we are playing CSI here. Right, the genetic disorder is caused by a dominant allele. Now remember, alleles, one from mom, one from dad. Those two genes together are going to be, and they are alleles. They are buddies. They are there for the same characteristic, okay? The genetic disorder is caused by a dominant allele. The findings of the scientists are represented in the pedigree diagram below. People, if you get a pedigree diagram, I almost want to give you permission to get up during the exam and do a little war dance around your chair and then sit down again. Because everything is given to you. All the information is in front of you. You just have to know how to use it. Pedigree diagrams are the bomb. So I want to make sure that you get 100% for any pedigree diagram that is given to you during the exam. So here we go. We have a square. Now we already know squares are guys and circles are girls. Why? Because we've got the curves and guys have got all those corners that they're so full of nonsense. You see, kids, there we go. I mean, that's not that I'm a feminist. And the guys, again, are killing themselves laughing. But yeah, us girls, we're rounded, you see. We're beautifully rounded. So there we go. There's our little girl and there's our guy. Okay, this is the first generation. Now, we go to the next generation. And this generation, we have a circle and square and a circle. Here we have a square, and this square gets together with a circle, and they have a square. So this is our second generation, and this is going to be our third generation. Remember, the generations run like that. So that's the first, this would be the second, and this would be the third, and so it goes down. So every straight row is going to be a generation. Here we've got the grandparents, the parents, and the grandchildren. Okay, we'd look at it like that. So we have now in our third generation, we have this little one and a square, and they are going to have a square, and this is X. Right, now the shading. Okay, here's the key. Square, square, circle, circle. This is an affected male, and this is unaffected. Okay, this clearly is an affected, because if that's an affected male, this must be an affected female. Okay, and an unaffected female. All right, now, oh, hang on, let's leave it at white. I just want to get rid of all of these so that they don't confuse us here. But I hope you understand what the generations are now. Okay. Okay, now, this is A, that's B, this is C, and this is D. Before we do anything, when you get this is what you're going to do. You're going to say, we're going to work backwards. It's called a back cross. Okay? You're going to work backwards. So you're going to start at the third generation, then you work to the second generation, and then you work to the first generation. Or you can just do one generation down. So look at this. Here we have an affected male, and we have an unaffected female. But they have told you here that the dominant gene, the genetic disorder is caused by a dominant gene. All right, so we need to find out which one is dominant, whether being affected or whether not being affected. Which one is normal and which one is the dominant gene, or recessive, and which one's the dominant. So how do we do this? Easy. We look at A and we look at B, and we look at the kids that they had. So A and B produce this child here, but they produce 2 boys that are fine, and they produce 2 daughters that are both affected. In order to produce a child that is affected, it means here and here, it means that each one of these parents had to have given the recessive gene. Now, I just want to see what letter. Do they say here the letters R and R? So R is dominant and R is the recessive gene. Okay, so A. In order for C to have a child and have a child there, but these 2 are fine, and this one definitely shows the sign. How on earth do we work it? So if we look at A must be heterozygous. And why do I say that? Because B is that. So that means this one and this one. And because there is only one dominant gene, we now have this one and this one. So now you're going to say, hang on, Kathy, how did you get that? Let me show you. In order for a dominant gene to show, if you remember the crosses we did last week, if I have, okay, let me get space here. If I do this, okay, and this is, sorry, and this is recessive. So I take homozygous dominant, which would be RR, and I take homozygous recessive, okay, that would be RR. Remember, for the recessive trait, you have to always have 2 alleles. In other words, it must be homozygous, it must be pure. Then your cross is going to be this. Okay, we also did a cross that looked like this. Now, clearly the parents were not the same because one is affected and one isn't. So we can do that. So this can either be small r, and we do this. Okay, so the child will be heterozygous. 50% will be heterozygous chance, and 50% chance that they will be homozygous. And that is how we came to this. Okay, how? Look at this now. We go further down. If this one is that, and this one is that, that's the only way this unaffected male could come about, because the unaffected male would be small r, small r. Okay, so there's your recessive trait. So basically, you work backwards, which means that every one of these white blocks are going to be homozygous recessive. So they are all, we write here, homozygous recessive. And this heterozygous. Okay, and in other words, any dominant place where that gene is, you are going to then have a shaded affected male and affected females. How do we know that this is recessive? Because these two here end up with the baby. Okay, now the questions. Using the letters R and R, so capital R for dominant and small r, which is exactly what we've done, it says state the genotype of individual A and D. We already know A is capital R, small r, okay, because it's heterozygous, and D is, what was D? Where's D? Here's D. D will be homozygous. Well, it is homozygous, but it will be small r, small r. When they ask the genotype, you write the letters of the genes, okay? So the alleles, what are the alleles? When they ask for phenotype, remember phenotype is what you physically are going to see. Then, what is the percentage chance of individual X having a genetic disorder? Now, here is individual X. So, if I do a heterozygous male and a heterozygous female, okay, we'll just do this quick. Heterozygous male and a heterozygous female, you are always going to have a 3:1 in the phenotype. Okay, so dominant, 3:1, and you are always going to have a 1:2:1 in the genotype. What do I mean by that? 1, 2 that are going to be heterozygous and 1 that is going to be homozygous recessive. Always. This is always. So here are 3 different crosses of the 4 that we did last week. Okay, so saying that, what did we have here? What was the percentage chance of individual X having the genetic disorder? Remember, X is going to have that genetic disorder as that, because there's a gene from mom and a gene from dad. Or they're going to have either the dominant gene from dad or the dominant gene from mom, and a recessive gene from dad and a recessive gene from mom. Okay, so what is the chance? It's going to be 3 out of 4 and 3 out of 4, because you're going to have 1 and you're going to have 2 that are that. Just like in our cross here. See? 1 that is homozygous dominant, and heterozygous is 2. That means it's going to be a 3 out of 4, and 3 out of 4 is equal to how much? 75%. And that's what they ask. They say, what percentage chance is there? It's going to be 75%. Because either that child is going to get 2 dominant genes, one from mom and one from dad, or 1 dominant gene from mom or dad and 1 recessive gene from mom or dad. Okay. If individual C, okay, so here's individual C. If individual, let's use another color. If individual C marries an unaffected male. So we're now going to have, let's put her in here. C marries an unaffected male. So we know the unaffected male is going to be small r, small r. State the possible genotypes of their offspring. So we've got heterozygous and homozygous. So and guess what? We did the cross just now. Heterozygous and homozygous recessive. So what is the chance here? They wanted to know the possible genotypes of their offspring. What are the genotypes? It's going to be 2 heteros and 2 heteros and 2 homos. So if these two have bubbas, okay, we are going to have is going to be 2 to 4 will be heterozygous, and I'm going to just write heterozygous here so you remember. And clearly this is a case of complete dominance, and we're going to have 2 out of the 4 possible combinations are going to be homozygous recessive. Okay, homozygous meaning pure, and recessive, they are the recessive genes, they are dominated. Hetero means different. Okay, homo is the same and pure. And if you remember those, you will be fine. Okay, now that's if we have complete dominance. Okay, remember Mendel's law. When you have complete dominance is when you have 2 parents with homozygous pure genes that are opposite, and they have a child. What do you end up with? You end up with the dominant characteristic or trait always showing in the F1 generation or the first generation. Always. The dominant gene will always dominate, and that is what you will see in the phenotype. Okay, now, okay, so that was that question. There was something I wanted to show. Okay, here. Let's just get, mind you, I've got space here. I want to show you this. You get complete dominance. Okay, when it is complete, your dominant gene will, your dominant trait will show in the phenotype. In other words, what you see, okay, in the homozygous dominant and heterozygous state. Okay, so what have I said here? What does this mean? It means that if it is, okay, let's go with the R's. If it is capital R and capital R, in other words, it is homozygous dominant, or if it is heterozygous, capital R, small r, then you will see the R trait. In other words, the dominant trait. Okay. When we have, and now I'm really going to run out of space. When we have incomplete dominance, and I'm not going to write the words out. When we have incomplete dominance, what happens then is you have a mixture. Okay, so let's go with the red, let's go with R and R. So we'll say R equals red and small r equals white. So when there is incomplete dominance, okay, R red and R equals white. So if it's that, we will see red. And if it's that, we will see red. Only when it is small r, will we see white. Why? Because that red gene dominates. When we have incomplete dominance, we're going to end up with a mixture, which means that when it is RR, we'll see red. When it is RR, which is homozygous recessive, we will see white. And this is the easy one. When it is heterozygous, we are going to see pink. Or if you cross black and white, fur on a rabbit, and we have incomplete dominance, we're going to end up, we have say a black dad and a white mom, and the two of them have little bunny babies. We can have babies that are going to all be what? They are all going to be little grey bunnies. That's if you have incomplete dominance. So incomplete dominance is also always an easy one to spot, because if it's got the dominant trait, it looks like the dominant trait. If it's got the recessive trait, it looks like the recessive trait. And when it's a mixture, so it's heterozygous, guess what? The babies are a combination of the dominant and the recessive. It's a mixture. Cows, you end up with a roan cow. So white cows and red cows, and they mix together, and you end up with this light browny, coffee sort of mushy color. Okay, so black, white, pink is incomplete. Complete, red, white. Complete, if it's red and white, white flowers only come about when there are two recessive genes. It's the only time it'll come through. Incomplete, it's a mixture. And then our last one, and now I'm not going to have space, is when we have codominance. Okay, and codominance, co means what? Co means together. So you have complete dominance, incomplete dominance, sort of mixture, and then you have codominance. So codominance is like a partnership. You have both partners equally put in the effort to make a success of a company. So, co-partners, here we have codominance. So both are equally dominant and equally recessive. They both come through when both those alleles are present, or both those genes are present. So when it is codominance, like, let's go with our flowers again. So we have red is, R is red, and we have W is white, okay? So that you see, I've now put that, so these are co-dominant. Then, what are we going to see? We're going to see when it is capital R, the flowers will be red. When it is capital W, the flowers will be white. And when we have a capital R, capital W flower, we are going to have a red flower with white markings. Okay, so if we think of cats, you get black and white cats. So black cat mates with a white cat, and they end up with a kitty cat that has got a fur that's black with white markings on it. Okay, codominance, blood groups. Look at this. In fact, you know what? Wait. We've got a question here. It was hemophilia. There's so many questions. Ah, here we go. Okay. This question was from Enoch, I think. Okay. Enoch said, human blood groups are controlled by multiple alleles. Okay, so let's get a new page. So we're looking at, I'm going to write human blood groups, and this relates to exactly what I'm doing now. Human blood groups are controlled by multiple alleles. Okay, this still hasn't got anything to do with codominance, but we're getting there. Okay, so in order for blood groups to be, you've heard, you've got A or B or AB or O. Okay, so what does all of that mean? It means this, that there are multiple alleles. So alleles that control blood group. In this case, there are 3. So let's see. We say here, first question is number 1, how many alleles? Okay, so in other words, how many alleles are going to control blood group? Well, that's pretty easy because you've got and you have, so what's that? It's 1, 2, 3. How many alleles? 3. That's for A, that's for B, and that's for O. Okay, and I'm just writing this here so that you understand. You can't. In an exam, if you have to list those blood groups, you have to write them like this, a line, okay, and, okay, so the little I is for O. And there must be 2 of them to make O because O is recessive. So here we go. We say, okay, I'll get to my code. Just hang on. It says, which 2 alleles are codominant in the inheritance of blood groups? Okay, well, this is exactly what I wanted to answer. So which, let me write here, which are codominant? Okay, it's going to be A and B. A and B are codominant. They are equally as powerful. That is why you have blood group A, blood group B. Guess what? Blood group AB and blood group O. So how does it look? And I'm going to just do that here so that you can see it with your eyes. I always believe if you see something, you can remember it. So these are our combinations. Okay, so remember, these 2 are codominant. So here are your combinations. You can have A, they will be homozygous dom. Okay, then, and this will still be shown as blood group A. Okay. Then you have, this is hetero, okay, and that's still going to show as, at least that will still show as blood group A. Okay, then we have the same for B. Okay, so this will be homo dominant. And it will be blood group B. And hetero, and it's going to be B, blood group B. Now, our last one, and I want you to be able to see all of them, okay, is, well, second last one, is homo recessive, and that's going to be blood group O. Now, we've said that A and B are codominant. So when I have A and B, it is going to end up being codominance. And we end up with A, B. Okay, there are blood groups. So you can have A, homo A, hetero A, homo B, hetero B, O, AB. They're your 6 blood groups. And then, of course, you've got the positive and the negative rhesus factor. So positive if you've got the protein, negative if you don't have the protein. So what do these questions say? It says, which 2 alleles are codominant? It's A and B. And you're going to write A and B like this. You write it just exactly like I've written it here. A line and an A and a line and a B. They are codominant. Then it says, okay, here's our question. We have to do this calculation. So they say, we have a man who is heterozygous for blood group A. Okay, so let's write this down. The man is heterozygous for blood group A. Okay, and he marries a woman. Let me write man here. A woman who has blood, oh, I should have just put BG. Blood group O. Now, the minute we see O, what do we know? It's the recessive gene. Okay, so the woman, what is hers going to be? It's going to be, and the fact that they tell us the man is heterozygous for A, we know that this is going to be, people, I hope you understand. Okay, it's just the way of writing it. Okay, so he's heterozygous. He's not pure A. He's heterozygous A, which means one allele is A and one allele is going to be O. So okay, now use a genetic cross to show the phenotypic ratio of the offspring. This is how you're going to make your marks. And this is where the fun comes in, because I love it. I love genetic crosses. So we go P1. Do not forget P1. That's half a mark. Okay? So we have P1, right? And what are the parents? So our phenotype, and everything I write here, you must write in an exam. So the phenotype is going to be blood group A times the woman, and the woman is going to be blood group O. Okay, then your genotype. Oh, I love genetics. I love it. Okay, so blood group A here is going to be, they tell you he's heterozygous, times O is going to be 2 little i's. Okay, then what do we have? We have meiosis. Meiosis in the guy's body and meiosis in the female's body. All right, and how does it all divide up? We end up with the gametes. And what are the gametes going to be? We're going to have one gamete, one possible gamete, and the other possible gamete times that little gamete, and then this one. There we go. There are gametes. Okay, now I need more space. We only need that really. Now we do our little Punnett square. And as I said to you, I like a Punnett square because you can see everything right in front of you. They're not all kinds of lines this way and lines that way and this way and that way. And by the time you know what's going on, it's like, no idea. So Punnett square is nice and clean and just like sorted. All right, so we take these two. So, okay. And these are your gametes. And here, so what have we got? We've got a heterozygous. Remember our crosses? A heterozygous versus a homozygous recessive. Let's just go back a page. Remember our crosses? Heterozygous and a homozygous recessive. That's exactly what we're working with here, people. So that's why I said to you, there are only 4 possible crosses. Okay? Therefore, number 1. Number 2, you need to know that blood your blood group is as a result of multiple alleles. And that's where codominance comes in. Okay? Because A and B are both dominant. The third thing is sex-linked. So anything that's sex-linked works on the XX and the XY chromosomes. So I want to do a question on that later. But okay, so here we go. We go back to our homozygous. We've got a heterozygous times a homozygous. So heterozygous here times a homo. Literally. And it's homo recessive. Okay. So we have it'll always be 50%, 50%. And this is your fertilization. Remember, each one of these, that one, this one, I didn't put the little dot there, and I didn't do the little one here. So this one here, that one here, this one here, that one there. Each of these is a 1 in 4 chances that that is going to be your combination, right? So even if you have 4 babies, or let's say a cat or a dog or anything in any cross, if they have, a dog can have 10 puppies. Each puppy is going to work on those 4 combinations. It could be, each puppy could be any one of those 4 combinations. The only time you have exactly the same gene combination and chromosome combination, the only time is if it is identical twins. Then you have 1 sperm, 1 egg, they fuse during fertilization, and then the egg splits into 2 separate entities. Okay? Then they're identical. Other than that, you can have 5, 10, 20 children. Each time you produce a child, it can be any one of these 4 combinations. Okay? Always the 4 combinations. All right? 4. And that's why when we do dihybrid, this is a monohybrid, 1 cross. When we do a dihybrid cross, in other words, you're crossing 2 characteristics, you have 16 combinations because there are 4 for the first, 4 for the second characteristic. What's 4 times 4? 16. Okay, now we have fertilization. So then with fertilization, we're now going to look at the F1 generation. So this is the first lot of babas. And that the offspring here, what is the combination of the offspring here? So the why do I always put a little dot there? It's because I've been doing all the other dots. Okay, so your F1 or your first filial generation, your genotype is going to be, so genotype, what is in the genes? Okay, the genotype is going to be 2 of the 4 are going to be there and there. A. So they're going to be your normal, and I'm going to put in brackets here, heterozygous okay? But the blood group is going to be A, but we'll do that now. Then we have 2 out of 4 chances that it's going to be II, which is going to be homozygous dominant. Sorry, recessive. Okay, right. And if we look at the phenotype, in other words, what are we going to be able to see based on what is in the genes? So this is what we will see. We are going to see 50% with blood group A. Because remember, you can't see the O. So 50% will have blood group A and 50% will have blood group O. I hope you all figured that out. I hope you got it. So where are your marks? Please, people, here. About to go for an ad break, but let's just quickly check. Where are those marks? I'm not going to use, can I use purple? Yeah, let's go with mauve. So F1, P1 plus F1 is equal to 1 tick. Okay? Your phenotype and your genotype gives you a tick. Your gametes and meiosis give you a tick. Do you have the correct gametes here? Yes, 2. Then you've already got your half a mark for that one. Your genotype, your phenotype. There are your marks. How easy is that? So I expect you to get full marks for a cross on blood types. Remember, blood types are they're multiple alleles because there are 3 alleles that are used, and you will only ever have 2. You can't have more than 2 in your body. So, or any of your nuclei on your chromosome, you can never have more than 2 alleles for blood. So you are either going to have A and O, or B and O, or A and B, or O. That's it. Done. Sorted. Okay? People, there you go. We're going to go, I need some water, I'm desperate. So we're going to have a quick ad break for 2.4 minutes, I think this time it's going to be, and I will see you all rocking and rolling with our next question.